Hello students, this is Dr. Ravi Sani. So today we are going to discuss about the hemostasis. It is defined as arrest or stoppage of bleeding when a blood vessel is injured. The injury initiates a series of reactions resulting in homeostasis. This is the definition of that homeostasis as arrest or stoppage of bleeding. Arrest or stoppage of bleeding when blood vessel is injured. So there are some reactions which occur in series and finally results in the hemostasis, which is called stoppage of bleeding or arrest of bleeding. So basically it has three stages. The first one is the vasoconstriction, a platelet plug formation, and the coagulation of blood. So in case of vasoconstriction, here the term is constriction. So immediately after injury, the blood vessels constricts and decreases the loss of blood from damaged portion. As due to the result of constriction, diameter will be decreased, and this will help in prevention of loss of blood from damaged portion. Usually, arterioles and small arteries constrict. Now come the second stage, that is platelet plug formation. As its name suggests, platelets aggregates together and form a loose temporary platelet plug or temporary hemostatic plug, which closes the ruptured vessels and prevents further blood loss. So basically, platelets are coming together, they are aggregating and forming a loose temporary mass, that is platelet plug or temporary hemostatic plug, which closes that ruptured part of the vessel, and it helps in prevention of loss of blood. The third stage is the coagulation of blood, in which fibrinogen is converted into fibrin. This fibrinogen is the plasma protein, which we read in the plasma proteins. The other proteins are albumin and globulin. The fibrin threads get attached to the loose platelet plug, which formed in the second phase, which blocks the ruptured part of the blood vessel and prevents further blood loss. So, in second stage, when the platelet plug was formed, in third stage, it got attached to the fibrin threads. These fibrin threads forms due to the conversion of fibrinogen to fibrin. And finally, there is a complete arrest of bleeding. These are the stages in uh, diagrammatic or in image form. This one is the stage 1, stage 2, and stage 3. Any injury to the blood vessel and damaged endothelium will result into the exposure of collagen, because we all know that collagen is the most abundant protein. This collagen exposure with will von Willebrand factor will result in the adherence of platelets to that collagen, and it will lead to the activation of platelets. These activation of platelets has three consequences. The first is the secretion of serotonin, which will result in vasoconstriction, secretion of ADP and thromboxane, which will result in aggregation of platelets. In the last, there is a platelet plug formation, and in the third stage, there is a formation of prothrombin activator, which will finally lead to the blood clotting. Now come to the stage which is coagulation of blood. Hemostasis is the actual broad term in which we had three stages. The third stage sometimes is referred as coagulation of blood or blood clotting. In this stage, coagulation or clotting in response to injury or trauma occurs. After the stoppage of bleeding, we saw a reddish brown scum which forms at the site of the cut or injury. After a period of time, a clot or coagulum formed mainly of network of threads called fibrins in which dead and damaged formed elements of blood are trapped. So in first stage, there was vasoconstriction. In second stage, it was platelet plug formation. In third stage, conversion of fibrinogen into fibrin. So this in this third stage, fibrin will form a network. This network will get attached to the platelet plug and will act as a patchwork to the damaged blood vessel. And in this network, the other blood cells also got trapped and helps in repairing or stoppage of bleeding. Normally, all the factors are present in the form of inactive proenzyme. Mainly, enzymes are in the state of inactive. Whenever there is a requirement of these enzymes, they got converted into the active form. These proenzymes must be activated into enzymes to enforce clot formation. It is carried out by a series of proenzyme enzyme conversion reactions. First one of the series is converted into active enzyme that activates the second one, which activates the third one. There is a continuous series. This process of coagulation is a cascade of enzymes. Cascade means there is a series. One enzyme will lead to the activation of the another enzyme. Activation of one factor leads to the activation of another factor. This is called the cascade theory or cascade of enzymes. Now, these are the further three stages of the blood coagulation. Hemostasis, we divided it into the three stages: vasoconstriction, platelet plug formation, and the third one is the coagulation of blood or clotting of blood. Here we are dividing this stage, third coagulation of blood, further into three stages. The first one is the formation of prothrombin activator. The second is the conversion of prothrombin into thrombin. And the third, it is the conversion of fibrinogen into fibrin. So these are the three stages of coagulation of blood, which is the third stage of hemostasis. In this image, we can see these three stages. These are the three stages. In this stage, there is a formation of prothrombin activator. In the second stage, there is a conversion of prothrombin into thrombin. In the third stage, there is a conversion of fibrinogen protein into fibrin. Now come to the first stage, which is the formation of prothrombin activator. For formation of prothrombin activator, we have two mechanisms, the extrinsic pathway and the intrinsic pathway. So the blood coagulation or blood clotting starts with the formation of substance called prothrombin activator, which converts prothrombin into thrombin. Its formation is initiated by substances produced either within the blood or outside the blood. That's why we have two mechanisms for formation of prothrombin activator, the intrinsic pathway and the extrinsic pathway. After formation of prothrombin activator, which converts prothrombin into thrombin. So in this image, we can appreciate both the pathways. Here we have extrinsic pathway and sorry, this one is the extrinsic and this one is the intrinsic pathway. So first we are focusing on the intrinsic pathway. So there is a endothelial damage which results into the collagen exposure. So this will result into the activation of factor 12 into 12A. This A shows activation, and this endothelial damage with these factors will help in conversion of this activated enzyme into active enzyme. This enzyme further lead to the activation of the another enzyme. Here we are having 9, factor 9, which is getting converted into the 9A. Same here, A represents activation. So first the 12 factor 12 got converted into the activated stage. This results into the activation of 11th factor. Again, this activated enzyme is again leading to the activation of the ninth factor. This ninth is leading to the activation of 10th factor. Finally, this is responsible for the formation of prothrombin activator. There is another pathway in which, due to collagen exposure, will lead to the platelets, phospholipids, and it could directly form prothrombin activator. In extrinsic pathway, it is quite simpler than the intrinsic. Same tissue trauma or tissue thromboplastin is leading to the glycoprotein or phospholipid exposure. This is this is responsible for the conversion of factor 10th. In the intrinsic mechanism, here we are having factor 10, and in case of extrinsic, in very early stage, there is a conversion of factor 10. Finally, with calcium or factor 5, there is a formation of prothrombin activator. So this is the first stage of coagulation of blood, which is titled as formation of prothrombin activator. In second stage, this prothrombin activator is responsible for the conversion of prothrombin into thrombin. In stage third, this thrombin is responsible for the conversion of fibrinogen protein into uh, fibrin. There are some other factors, calcium, and here we are having a formation of loose strands of fibrin. This fibrinogen is getting converted into the fibrin, and these fibrin threads will form a network or mesh-like network in which platelet plug got attached, and the other cells of blood also got trapped into that, which helps in arrest or stoppage of bleeding.